Can we clone a dinosaur? Many of us have wondered what the world would be like if we even had one dinosaur. So much is not known about them, and the potential to have even one would provide so many scientific breakthroughs. In this video, I'm answering, can we clone a dinosaur? Should we clone a dinosaur is an entirely different question. A very popular topic in paleobiology is a recently discovered Tyrannosaurus femur that was recently unearthed in Bozeman, Montana. Now, the fossilized bone had near perfect ligaments, collagen, and even blood encased inside of it. But did it have DNA? The fossil was taken back to Montana State University, where the incredible overturning discovery was refused by the university's paleontologists to be tested for DNA. So before we go further, I would like to explain what DNA really is. DNA is genetic material that forms into a double helix. The double helix reacts with histone proteins, which are proteins that bind to the DNA. They wrap the double helix around themselves, and when many of these proteins come together, they form the DNA into a chromosome. The chromosomes are stored in the nucleus and the mitochondria of the cell. If there was DNA in the Tyrannosaur tissue, it would be free-floating, considering histone proteins decay in as little as 5 days after the death of the organism that they belonged to. So what does this have to do with our Montana fossil? The Creation Institute in Kentucky was granted permission to study this specimen as long as all samples were returned to Montana State University when the testing was complete. The scientists took a sample of blood from the femur and injected histone proteins into the specimen. If there was DNA in the blood, the proteins would react, causing the DNA to wrap around the proteins. The reaction was present, and DNA could be clearly seen wrapping around the proteins through a microscope. They had got what they were looking for: dinosaur DNA. Until only recently, a clone could not be created simply with a pile of DNA. However, Dr. Jerry Yang from the University of Connecticut recently published his findings in the journal titled Nature Genetics. His research revealed that clones could be created using fully differentiated white blood cells. Before this discovery, mature differentiated cells could not be used in the cloning process. But the blood of the Tyrannosaurus that was found in Montana had all that was needed for this aspect of cloning to take place. Except one thing. What would be needed now was somatic nuclear transfer. This is the aspect of cloning done by Yang and his team. This is when the nucleus of a cell of the animal that is planned to be cloned is injected into an egg cell whose nucleus has been removed. Unfortunately, there are no Tyrannosaur egg cells, only the cells within the femur that were discovered. However, if the animal egg cells used are close enough in relation, they can be acquired from a different species if needed. The skull and overall shape indicates that Tyrannosaurus, and all dinosaurs for that matter, belong to the Archosaur family. Other members of this family are the modern crocodiles and alligators. Since dinosaurs are so closely related to crocodiles and alligators, an egg from one of these creatures should theoretically work to successfully clone a Tyrannosaurus. The DNA from the white blood cells of the Montana Rex can, with our modern technology, be inseminated into a female crocodile egg. The mother croc's uterus would be the closest version to the uterus of an adult Rex and should suffice in the development of the dinosaur. With technology increasing dramatically, it only may be a matter of time before John Hammond's famous quote becomes a reality. Ladies and gentlemen, welcome to Jurassic Park.